Hi there, and welcome to the second of 5 segments in which I'll cover lecture material on the nervous system. So in this second segment, we will cover the slides in part 1 of 2. We will cover slides number 83 to 111, and that's the end of part 1. So in the first segment, in the previous lecture, I covered points number 1 through 6 up to structure and function of neurons. In this second segment, I will cover points 7, 8, and 9, and we will pick up with number 7. Number 7 is why the next neuron may or may not fire an action potential, and we'll learn about postsynaptic potentials. So to give you a little context around this, we had previously been talking about how neurons communicate at a synapse. And we'll continue to talk mostly about chemical synapses. And I hope you remember that at a chemical synapse, the presynaptic neuron releases a neurotransmitter. That neurotransmitter diffuses across the cleft and binds to neurotransmitter receptors on the postsynaptic cell. And those neurotransmitter receptors are ligand-gated ion channels, and when they open, they will allow a particular ion to either enter or leave the cell down its electrochemical gradient. That will then cause a change in membrane potential in the postsynaptic cell, and that change is called a postsynaptic potential. So a postsynaptic potential is how membrane potential of the postsynaptic cell changes. A postsynaptic potential can either be depolarizing, the inside of the cell becomes less negative, or hyperpolarizing, the inside of the cell becomes more negative. And what makes the difference is what kind of ion channel is opened by the neurotransmitter. So you're familiar with this image. It's a chemical synapse, and let's see if you can answer this quiz question. What happens to membrane potential in the postsynaptic cell when sodium enters the cell? Just as is shown here. Those are ligand-gated sodium ion channels. The neurotransmitter binds to them, causes them to open up, and you remember that there's more sodium outside the cell than inside the cell. So when those channels are open, sodium enters. How does that affect the membrane potential of the postsynaptic cell? That's right. Membrane potential will start to depolarize. If that postsynaptic cell was at resting membrane potential, negative 70 millivolts, that influx of sodium is going to cause it to go to, let's say, negative 69 millivolts, negative 65 millivolts, negative 60 millivolts. That's a depolarizing a depolarizing postsynaptic potential. That's also known as an excitatory postsynaptic potential, or an EPSP, because that's a bit of a mouthful. Even EPSP is a mouthful. An excitatory postsynaptic potential brings the membrane closer to threshold. As the postsynaptic cell moves from negative 70 millivolts toward negative 55, that cell is closer to firing an action potential. This is why it's called an excitatory postsynaptic potential. All right, here's a different quiz question. We have to pretend now that these are not sodium ion channels, but rather they are potassium ion channels. So flip that in your mind. Now the neurotransmitter is opening up ligand-gated potassium ion channels. And you remember that there's more potassium inside the cell than outside the cell. And so when those channels are open, potassium is going to leave the cell by facilitated diffusion. And what happens to membrane potential in that case? Yes, you got it. Membrane potential will hyperpolarize. That postsynaptic cell was at negative 70 millivolts. That was its resting membrane potential. Then a bunch of potassium left the cell and took their positive charges with it. And now that membrane potential is maybe negative 72, negative 75, maybe it's negative 80. So now the membrane becomes temporarily hyperpolarized. And that is called an inhibitory postsynaptic potential, or an IPSP. We say it's inhibitory because now that cell is further away from the threshold, and even less likely to fire an action potential. So we say that that postsynaptic neuron is now inhibited. It's like the presynaptic neuron went, shh, don't fire an action potential. Because it's hyperpolarized, it's less likely to reach threshold. All right. So we've been looking at a very simple system where one cell communicates with the next, but the reality is most neurons connect with many, many different cells. Most neurons have many synapses on their dendrites and their cell body, and a single postsynaptic cell can be stimulated by thousands of presynaptic neurons. And each of those presynaptic neurons will release a particular neurotransmitter that opens up a particular type of ion channel and causes either an excitatory postsynaptic potential or an inhibitory postsynaptic potential. Let me show you, I hope the next slide is a picture of a neuron that is connected to many other neurons. So you get a sense of how many inputs there can be, incoming communication points that there can be on a postsynaptic cell. Yes. So here we are looking that the major white blob there is the cell body of the postsynaptic neuron. And all of those tiny sort of footed structures are synaptic terminals, synaptic end bulbs of presynaptic neurons. So at each one of those cell-to-cell junctions, a neurotransmitter is secreted and by the presynaptic cell, and the postsynaptic cell has ion channels present in that part of the plasma membrane, ligand-gated ion channels. So the postsynaptic cell can receive a message from the presynaptic cell at all of the points that you see here. We'll go back to a simpler version where we have one postsynaptic neuron, and this postsynaptic neuron is only receiving information from just 3 presynaptic neurons. And so some of those presynaptic neurons can cause excitatory postsynaptic potentials, and the others cause inhibitory postsynaptic potentials. So what is that postsynaptic neuron to do? Is it going to fire an action potential or not? This is a fantastic image for illustrating the various influences that different presynaptic neurons can have on a single postsynaptic cell. So the postsynaptic cell is sort of in that pinky beige color, and we have 4 different presynaptic neurons. One is green, blue, black, and red. Those are 4 different presynaptic neurons, and they each release a particular neurotransmitter. The postsynaptic cell at that junction has a particular type of ligand-gated ion channels, and for each cell-to-cell communication, the postsynaptic potential will either be excitatory or inhibitory. So in the little sort of grayish blue squares, we can see the outcome of communication from that particular presynaptic input. So the green one up top, we can tell by the gray square and the little plus sign that when that green presynaptic neuron releases neurotransmitter, it causes a local excitatory postsynaptic potential. So on the postsynaptic cell, the plasma membrane in that region depolarizes. And now let's look over at the red the red presynaptic neuron down below. When that presynaptic neuron releases neurotransmitter, it causes an inhibitory postsynaptic potential. And we can tell that by looking at the little gray square and also the minus sign. So in that region of the membrane, the postsynaptic cell will hyperpolarize. And then notice on the right where it says action potential, question mark, that question mark reflects, well, we don't know if the postsynaptic cell is going to generate an action potential at that trigger zone. It depends on whether or not that trigger zone reaches threshold. So we are talking about summation. Summation is the integration of multiple presynaptic neuron inputs on a postsynaptic cell. So that postsynaptic cell will sort of combine all of the presynaptic inputs. And the greater the number of excitatory inputs, the greater the likelihood of an action potential. The greater the number of inhibitory inputs, the less likely an action potential becomes. So here's the question. Should Nellie the neuron fire an action potential? Well, it depends. So during summation, the effects of neurotransmitters released by a single presynaptic neuron or multiple presynaptic neurons, the effects of those neurotransmitters build on each other in the postsynaptic cell. And during summation, there will be some outcome in the trigger zone. What is the change in membrane potential that reaches the trigger zone? That is the important outcome of summation. So, when will the postsynaptic cell actually generate an action potential? Complete this fill-in. Summation will only result in an action potential in the postsynaptic cell when the trigger zone depolarizes to threshold, which is negative 55 millivolts, in our classic example of a neuron. In reality, different neurons have different thresholds. We'll always use negative 55 in here. All right, so that trigger zone, which is also known as the axon hillock, if those presynaptic inputs can get the membrane to depolarize to negative 55 at that region of the membrane, then an action potential will be generated. And remember, action potentials are all or nothing. When they happen, they will propagate the entire length of that axon to the synaptic end bulbs of the postsynaptic cell. All right, so there are three possible outcomes of summation. Let's think about those four presynaptic cells that we see, the green, the blue, the black, and the red. Let's imagine the various patterns that they can fire, the various patterns that they can deliver neurotransmitter. And if those neurotransmitters, if during summation, the trigger zone does not reach threshold, but is excited, that's called, number one, a subthreshold EPSP. So let's imagine that presynaptic cell green and presynaptic cell blue both cause excitatory postsynaptic potentials, but it's not enough to get the trigger zone all the way to negative 55. Let's say we get the trigger zone only to negative 60. Then no action potential will occur, but we can think of the cell as being primed. It's a little closer to threshold, so it will be easier. Let's say if now the black presynaptic cell releases its neurotransmitter, then it makes it far more likely that that postsynaptic cell will fire an action potential. And that's outcome number two. Outcome number two is called a threshold EPSP, or a threshold excitatory postsynaptic potential. And that just means that the threshold is reached at the trigger zone and an action potential will be initiated. And then possible outcome number three is an inhibitory postsynaptic potential. Let's say presynaptic input, the red one, fires a bunch and a bunch of that neurotransmitter is secreted and the postsynaptic cell responds and it is hyperpolarized. So now the postsynaptic cell, it goes from negative 70 to, let's say, negative 80. That's an inhibitory postsynaptic potential because the neuron is inhibited. It is less likely to fire an action potential. All right, signal and coding. How is it that your brain can distinguish between a love tap and a spank, right? Both of them involve touch receptors in the skin, sending signals to your central nervous system. Why do they feel very different from each other? All right, so if all action potentials are the same size, and all action potentials are all or nothing, they happen or they don't, how does your brain distinguish between a small stimulus, like a gentle breeze on your face, versus a large one, like the blow dryer on your face? All right, here are some funny pictures. We have a normal handshake versus somebody shaking your hand and they clearly have something to prove. And then we have a gentle caress versus a smack. All right, so how does our nervous system encode the intensity of a particular stimulus? Where the gentle caress is a low intensity stimulus and the smack across the face is a high intensity stimulus. Obviously, your brain can tell the difference. And so that intensity is encoded two ways. The first one is the frequency of action potential. So think about the touch receptors that are in our skin, the Meissner's corpuscles, the Pacinian corpuscles, the Merkel discs, etc. And it's okay if you've never heard of those things. There are a variety of sensory receptors in our skin. And in response to the smack across the face, they are going to fire action potentials in rapid sequence. Signal, 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 signal. Versus that gentle caress. Signal, signal, signal. So the second way in which the intensity of the stimulus is communicated to your central nervous system is by the number of sensory receptors that respond to the signal. So we see that image of skin there. And there are a great variety of sensory receptors. And let's take a look at, for example, see that Pacinian corpuscle all the way at the bottom? That is a mechanoreceptor. It's one of our touch receptors, and it's very deep in your dermis. So it's not near the surface of your skin. It's further away from the surface of your skin. And a gentle caress may not activate the Pacinian corpuscles, but a smack sure would. So in the gentle caress, fewer sensory receptors will be firing action potentials. And in the smack across the face, pretty much all of them will be firing action potentials. All right, I hope you have had well, can't really say it's a pleasure, but I'm sure glad that they exist. Maybe you have personal experience being treated with a local anesthetic, and I hope you never experience a neurotoxin. However, anesthetics can be considered a neurotoxin. A neurotoxin is something that disrupts the function of neurons. So let's think about local anesthetics. And for most people, this has come up in the context of the dentist's office. That's where most people experience local anesthetic. So let's say you need to have a cavity filled and the dentist will inject something into your gums and that causes your mouth to become numb. And it makes the cavity filling process a lot easier, a lot less unpleasant, because you become numb. You don't sense, you don't perceive any painful stimuli arising from your mouth as the dentist is drilling a giant hole in your tooth. So how do these local anesthetics work? Well, it's a type of molecule, and there are many different local anesthetics, and they're all a different molecule, but what they all have in common is they bind to some type of ion channel and prevent it from opening. And maybe you've noticed you can recall the effects of a local anesthetic. Not only does it block the sensory signals, and that's why we use it, right? Because we don't want to perceive the pain. So it will block the sensory signals, say traveling from your tooth to your brain. Those sensory neurons are not functioning under the effects of the local anesthetic, but it also blocks motor signals. So maybe you've come out of the dentist and half of your face is sagging, or you try to talk and you speak in a funny manner because your muscles are paralyzed. Or maybe you've tried to drink through a straw and it doesn't really work because those muscles are temporarily paralyzed. The motor output is also inhibited. It can't get those motor signals, can't get from your central nervous system to the muscles in your face in this case. So we have both numbness and paralysis. Here's a way to visualize how a particular local anesthetic works. So on the left side of the image, we see normally functioning voltage-gated ion channels, right? And depending on membrane potential, the ion channel could be closed or the ion channel will open at a different membrane potential. Well, when we inject a local anesthetic, the anesthetic molecules can bind to and block the voltage-gated ion channel, whether it's a sodium ion channel or a potassium ion channel. It actually doesn't matter. You need both functioning ion channels in order to generate an action potential. So let's imagine that that's a voltage-gated sodium ion channel, and now it can't open. Well, you can't have an action potential if your voltage-gated sodium ion channels can't open. Oh, that little rascally guy. All right, neurotoxins. So animals have nervous systems. Animals have nervous systems. Other types of organisms don't have nervous systems, at least not in the same way. So if you think of fungi and bacteria and plants, they don't have neurons. And because animals have nervous systems and lots of other animals eat animals, many animals have sort of weaponized chemicals that act as neurotoxins. And these neurotoxins are obviously a product of natural selection, and they can help protect an organism from predators. And so here are just two examples. Nature is full of different neurotoxins. These are just two examples. Tetrodotoxin is produced by the Japanese pufferfish, and it works by binding to and blocking voltage-gated sodium ion channels. And perhaps you're familiar with a sushi specialty that is pufferfish, and a sushi chef requires additional training in order to be permitted to prepare pufferfish, because there is a risk of injury or death to your sushi customer. And my understanding is that the goal here is to prepare the meat, the flesh of this fish, where it includes just a little bit of the toxin. And I often ask in class if anyone has any experience eating pufferfish. And the general consensus that I've gotten over the years, for the few students, there's usually one or two, sometimes there's none, but there's often one or two students each semester that have tried it. And I've drawn two basic conclusions. Number one, it's not particularly delicious. It's not delicious, but you pay more for it. And it's not that the fish, the meat, is any more delicious. It's okay, is what I get. But sometimes it can cause a tingling sensation on the tongue. And so that's evidence that the tetrodotoxin, a little bit of it is present, and it's disrupting the sensory function and the motor function of the neurons serving the tongue. And then down below, we see a poison dart frog. These are beautiful little frogs that occur in Central and South America, and there are many different species, and they are all pretty much brightly colored. And they make batrachotoxin, and they secrete that in their skin. So let's say a snake comes along, and they're little. These are little frogs. A snake comes along and thinks that this frog will be a tasty meal. Well, the snake, the mucosa of the snake's mouth will experience this batrachotoxin, and it will cause the snake mouth to become paralyzed, presumably numb, but definitely paralyzed. And so that allows the little frog to flee. This was an interesting piece of research that was published back in 2017. So some scientists wanted to know why frogs don't poison themselves. What makes the frog nervous system immune to the neurotoxin that it produces? And so it turns out that frogs, this particular species of frog, has a genetic mutation, and it's a change in 3 of the amino acids that make up the receptor in the nervous system, and it changes the shape of the receptor just ever so slightly so that the toxin cannot bind to or block the frog's own receptors. So this research gives scientists information about the toxin that could eventually be helpful in designing drugs such as pain relievers and drugs that are used to fight addiction. We can manipulate nervous systems using technology that Mother Nature, natural selection, has invented. All right, bungarotoxin. Bungarotoxin is a neurotoxin produced by a particular group of snakes. This is a banded krait, and it's found in southern Asia. And bungarotoxin, when it's injected into either a would-be predator of this snake, or the snake is also a hunter, it will use bungarotoxin to kill its prey as well. So when that bungarotoxin is injected into either a predator or a prey, that organism will become paralyzed because it blocks acetylcholine receptors. And you know about acetylcholine. Acetylcholine is required for transmission at the neuromuscular junction. So the motor neurons might be secreting acetylcholine, but the muscle cells are unable to respond. There's no depolarization of the muscle cells because those acetylcholine receptors are blocked. So it induces paralysis. And a good dose of this will lead to, in a human, it will lead to respiratory arrest. So the diaphragm becomes paralyzed. And here's where I tell you a very sad story. Do you see the initials on this snake, JBS? So that's Joe Slowinski. And very sort of between college and graduate school, I had an internship in Louisiana, which is where I am right now. I had an internship in Louisiana, and I have a history in herpetology. I've done research on turtles and lizards. I was never a snake person, but I love them, but I never did any research on them. And so I intersected with Joe at a university in Louisiana, and Joe was a crazy snake guy. And in herpetology circles, or even if you're into any, ology, like ornithology or ichthyology, everybody knows the crazy snake guys. And by crazy snake guys, I mean they are infatuated with venomous snakes. Not just all snakes, but they really love the venomous snakes. And by the way, I've never met a crazy snake woman. They must exist. I've just never encountered one. So you see these men at the herpetology conferences, and they're in the lobby in between sessions, and they're pulling up their pant legs, they're pulling up their sleeves, they're showing each other scars, and they're talking about how many times they've been hospitalized because they were bit by a venomous creature. And they were bit not by accident, but because they were handling these venomous creatures. And so that was Joe, very passionate guy. And Joe, when he finished his PhD, he got a dream job. Joe became a curator of herpetology at the California Academy of Sciences. And so Cal Academy sends expeditions out all over the world to catalog the biodiversity in some remote corner of the earth. So the people who are native to the area are familiar, obviously, with the plants and the animals and the insects and the fungi, etc. But many of these species are unknown to science. And so Cal Academy would send these research expeditions, and there would be botanists, there would be entomologists, there would be mycologists studying fungi, herpetologists, ornithologists, and it was their mission over several weeks to catalog as much of the biodiversity as possible. And often on these expeditions, local youth are employed. The kids know where the beetles live. It's the kids who know how to catch the lizards or where the lizards are. And so you just, you pay the kids, a few coins, and you say, oh, please go and get me all the worms you can find. And that's really helpful. And so the story goes, one morning, Joe, comes out of his tent and somebody hands him this snake in a bag. And that's what you do with a snake. You put it in a sack, like a pillowcase, and you tie it off. So the snake can move around freely and the snake isn't going to get injured. So a snake in a bag, that's normal operating procedure. And so Joe, who is a super duper trained professional, you know, sticks his hand in the bag, knowing totally what he's doing, but he gets bit. And he gets bitten by this snake. And he tells his team, okay, send a runner for help. Send a runner to the next village for help. I'm going to go lay down in my tent. Eventually, I will become paralyzed and you will need to breathe for me until help arrives. And that happened, and help never arrived. And his teammates, 3 or 4 people, took turns breathing for him, exhaling into his respiratory system for 24 hours. And they describe their experience as it feels like their lungs were shredded. It takes an incredible amount of effort to expel air into somebody else's respiratory system over such an extended period of time. And unfortunately, Joe died. I will counter that with a happier story later on. So this brings us to the end of segment 2 of 5 segments on the nervous system. I'll see you next time.